a big welcome. Thanks very much, Lloyd, for the kind introduction. Can everyone hear me okay? Excellent. So welcome to the last July lecture. I want to begin by thanking David Jamieson for the invitation to come and talk to you tonight. It's a real honour for me to be part of the 50th anniversary of these July lectures. And these are important events for the university, and we thank all of you for making this event a huge success. And so over the last 3 weeks, we've heard from some keynote speakers on some of the big areas in physics to do with particle physics, cosmology, and general relativity. And so in tonight's lecture, what I want to do is tell you about some of the work that's happening in the area of quantum mechanics. And in particular, how this work is leading to new quantum technology that we're now taking advantage of. And what I hope to demonstrate to you tonight is an example of quantum technology here. And so the title of my talk tonight is Quantum Mechanics and Biology, and what are the prospects? And so you may initially think, what on earth has quantum mechanics got to do with biology? How are these two linked? And so I hope over the next hour or so to be able to convey where we think these two areas of science can come together. And in the spirit of these 50th anniversary lectures, I want to take a look back at some of the significant milestones that have happened in these two areas of science, and then a little bit into the future about what we can expect in terms of new quantum technology and how this technology can be applied to understand and learn new things about biological systems. So that's where I hope we end up in the next hour or so. And so I wanted to begin by putting up a slide that just represents the world as we know it, the world that we're very familiar with. So I've got some everyday objects here. So we've got things like tennis balls, radios, birds and the bees. And this is on a scale bar here. This is a logarithmic scale. So every division on that scale bar goes down by a factor of 10. Okay, so we get down to things like the width of a single human hair, which is a couple of hundred micron. Here is pollen. And if we keep going down in scale, we get down to the single cell level, which is around 20 micron. This is 20 millionth of a metre. And if we keep going down, we've got red blood cells, bacteria, and viruses down in the nanometre range. Now, we can understand a vast majority of this world thanks to the efforts of people like Newton and Maxwell. And they gave us these mathematical theories to allow us to predict the behaviour of particles and waves. And we can do that very well in this classical world over an enormous scale range. Okay, but as we reduce the size of things, and as we look at fewer and fewer molecules and atoms, it turns out in this world, okay, the behaviour is very different. And so we need a new set of mathematical laws, a new description which describes the mechanics and the behaviour of molecules, atoms, and the subatomic world. And it turns out this is what quantum mechanics is describing. And so quantum mechanics was founded by some of these people here. So this is Niels Bohr, Einstein, and in particular Schrodinger, who we'll hear a lot about tonight. And so now we have these mathematical descriptions to describe this behaviour, and this has been tested again and again and is extremely accurate, and it's one of the most profound theories in science. But if we go back a little while into the 1900s, this was the idea of our atom. Okay, this was the so-called Bohr model. And the idea of an atom back then was you had a nucleus and you had an electron that orbited that nucleus. And so this was a very classical picture in some sense. This is like a satellite orbiting the Earth. And Schrodinger, with the Schrodinger equation, said this is not how we should think about an electron at all. We need to rewrite these physics textbooks, which still show this image here. And we should think about the electron as being a probability, a cloud of probability, if you like. Okay, so we can't know exactly where the electron is, but we can know the probability of finding an electron in a particular space. And so depending on the state of an atom, the probability distribution will change. So if it's in the ground state, the probability of finding the electron will be described by areas coloured in red here. And if we excite the atom, that probability distribution will change. And now we're more likely to find it in the yellow regions, for example. Okay? And this idea then brought up interesting questions about what is the electron doing? How can the electron occupy all of this space at one time. And this brought about the concept of superposition, the idea that the electron is occupying many different places at once. And this is a classical kind of example to illustrate what superposition means. So if you have a look at this image here, okay, for a little bit, and I'll take a little straw poll. Okay, so how many of you here, put your hand up if you see a rabbit in that image? Okay, and now put your hand up if you see a duck. Okay, so we've nearly got 50-50, okay? So there are two possibilities, there are two states that this picture is representing. It's representing both a duck and a rabbit at the same time. And it's only when we try to interpret this image, when we try and observe it and identify what it actually is, that it projects into one state or the other. It's either the rabbit or the duck. Okay? But while we're not observing it, it's in this superposition, if you like. Oh no. It did. Quantum superposition. There we go. So this is like a classical picture of, or the idea of what superposition is. But can we actually measure it? How do we actually prove superposition? And so what I want to talk to you about is one of the most elegant experiments that's been done, and it's called the double slit interference experiment. And it goes something like this. So this experiment was proposed by Young. It was called Young's double slit experiment back in the 1800s. And originally this experiment was designed to prove the wave-like nature of light. Okay? And it goes like this. So we have waves of light, so coherent light of a particular colour that's moving in phase, and these days we would put something like a laser that's incident on two very narrow slits. Okay, so the light is incident on these slits, and when the light goes through the slits, what happens is it emerges as if there are two new sources of light. And these light sources then radiate outwards. And what you have is there's areas in which one of the light waves will interact with the other light wave from the other slit. And areas where a peak from one of the light waves meets a peak from the other light wave will add together, and we call that constructive interference. And then there'll be positions where a peak of one wave will add with a trough of another wave. We call that destructive interference. We won't see anything. So if you get a screen now and put that screen a certain distance away from these slits, you'll observe what's referred to as an interference pattern. And so areas where we've got constructive interference appear bright, and then we have destructive interference, which is dark, and then we get these series of bright, dark, bright, dark. Now, anytime you see an interference pattern, you should think waves. Okay? Waves are responsible for these interference patterns. We see this was a video I took down at Altona Beach the other day. There was two-wave interference going on down there as well. Okay? Happens in water waves, happens with light. And the nice thing about this experiment is if you want to compare whether something's a light or a particle, if you think about if we put particles through this experiment, if I get a bucket of sand, pour it on these two slits, what will I see? I'm just going to see two piles of sand, okay? And I'm going to get two stripes, basically, on my detector. So this single experiment allows us to determine whether the source of the irradiation is behaving as a wave or a particle. Okay, so this was back in the 1800s. In the 1960s, people thought about, what if we do this experiment with electrons? Instead of light, let's throw electrons at these two slits and see what happens. Okay, so here's my screen. Every time an electron hits that screen, I'm going to represent that with a little blue dot. Okay, so when we do that experiment, this is what we see. So if we integrate this many, many times, gradually what you build up, and I hope you're familiar with it from the previous slide, is what's called an interference pattern. So we've got a series again of bright, dark, bright, dark. Okay? Now this was no surprise. So Louis de Broglie, back in the early 1920s, wrote a thesis about a property called matter waves. So he argued that any matter should have a wavelength associated with it. So wave-particle duality extends beyond light to all matter. Okay? So for Louis de Broglie, this was an example or proof of his theory, if you like. And the question was then, well, what's interfering? So these electrons go through the slits. What happens then? Do the electrons then interfere with one another? And that's what gives rise to the interference pattern? or is there something more complex going on? Okay, so they refined this experiment. And so then they said, okay, let's take an electron gun and fire a single electron, one at a time, through these double slits. And what are we going to see? And so these group of Italian scientists here performed the experiment, and Richard Feynman, a famous quantum physicist, had this quote, a very nice quote about this experiment. Okay, so let's do this. So one at a time, single electrons, are they going to go through this slit or this slit? Are we going to see a particle pattern, or are we going to see an interference pattern? Okay, that was the question. And when you do the experiment, what's interesting is that we're back to this interference pattern. Okay, this interference pattern from a single electron travelling through two slits. Okay, how can that be? That's quite odd. You would have thought that the electron would either go through the left or the right slit. Okay, but here we're getting this interference pattern. Something is interfering and producing this set of fringes again. So this was a really profound experiment. Okay, so let's try and get a little bit clever now. So let's try and understand where the electron's going. Is it going through this slit or is it going through this slit? So let's get a detector. Okay, and this detector is going to tell me when an electron goes through a particular slit, it's going to fire off a red beam of light to say, yep, the electron's gone through the slit on the right, or no, it hasn't gone through the slit on the left. Okay, so let's do this, put this detector there. Here's our screen. And we're going to run the experiment again, and we're going to look at which slit it went through. So my detector's getting clicks. Okay, I'm getting hits on the screen. But what you notice now is this interference pattern's gone. Okay, now the electron as if it's been a particle the whole way through this experiment. So how can this be? Just the act of putting a detector here and asking a question of which slit that electron went through has now totally changed the outcome of this experiment. Okay, just the act of measuring something. And so, okay, maybe this detector has introduced something funny. Maybe I'm some sort of magician and I'm tweaking the electrons in some way. So I'm going to leave the detector there. I'm going to go over to the PowerPoint and I'm going to turn the detector off. Okay, so the detector's still in place. We're still going to run the experiment. We'll just turn the detector off. Okay, we run the experiment again. And lo and behold, we get our interference pattern back again. Okay, so this is one of the amazing things about quantum mechanics. And so the explanation behind this interference pattern is that this single electron and its wave-like properties travel through both slits at the same time. It's this principle of superposition. It's going through two slits at the same time. A very strange concept. And then on the other side of the screen, it's the wave-like properties that have gone through these slits are interfering to produce this interference pattern that we see on the screen. So a very nice and elegant way to see superposition. And so in this one experiment, this one very cool experiment, we can prove the wave-particle duality that exists between electrons. You can do the same thing with light. We see the principle of superposition, so things can be in two places at once in the quantum world. And we can see the act of measurement. The act of measuring something, trying to measure a quantum state, impacts the outcome. Okay? And finally, for this interference pattern to occur, all of these sources need to remain coherent through this whole experiment. And more recently, people have even gone further, and instead of firing single electrons, they're throwing molecules at these slits. Okay? And again, Louis de Broglie would say, well, yes, there's a wavelength associated with molecules. And so people have thrown what are called buckyballs. So these are molecules which are made of 60 carbon atoms. Throw these at two slits and you'll see this interference pattern again. Okay, an amazing thing. So this is quantum superposition. All right, and this is one of the things that we look for and we now take advantage of in the new quantum technology that we're developing. The other quantum effect I want to talk about is something called quantum tunneling. Okay, so quantum tunneling was also predicted in this mathematical theory from Schrodinger, and this effect can be illustrated here. So imagine you've got some sort of barrier, some sort of energy barrier. I've got an object on one side, and this object wants to get to the other side. Now in the classical world, the way that we would do that is we pick up the line and we throw it over the wall. Okay, we need to give that line enough energy to get thrown over the wall, and that's the only way the line's ever going to make it there. In the quantum world, that's not what happens. Okay? In the quantum world, this is what happens. Okay? So in the quantum world, because things are dealt with in terms of probability, and because if you make this barrier narrow enough, there will be a certain probability that this object can exist on the other side of the barrier. If you leave it interact with that barrier for long enough, there will be an instant where the object appears on the other side. Okay? Although it hasn't got enough energy to get over the barrier, it can what we call quantum tunnel through that barrier. Okay, now this may sound like some sort of crazy theorist's dream, but this happens every day. This is one of the processes that powers the sun. So in nuclear fusion, what happens is protons come together. So you bring in two protons together, two positive charges. They want to repel each other, and that introduces this energy barrier. And every now and then, if you can get this interaction to last long enough, one of these protons will tunnel into the other, and you get nuclear fusion. Okay, and that's responsible for the sun shining. So this happens. Okay, so the question is, so I wanted to talk about those two effects, and then ask the question: does superposition, does quantum tunneling have anything to do with biology? So the title of the talk was about how quantum mechanics and biology are linked. And so we're certainly not the first people to think about that, and one of the first persons to think about it was one of the founders of quantum mechanics, so Erwin Schrodinger. And so in 1944 he wrote this book. This is a beautiful book, and I recommend you read it. It's a very short book, it's maybe 30 pages, and it was published based on a set of public lectures he gave at Trinity College in London in 1944. And what Schrodinger did in this book was try to address the pressing biological question at the time of what is the basis of heredity. So heredity is the way information is transferred from one living system to another, from one generation to another. And what Schrodinger argued was that heredity is too perfect. And it's too perfect to be described by classical mechanics. And his argument was that when we inherit the genes from our parents, we do so with extreme accuracy. And so when we inherit our genes, we might get an error of 1 in 1,000 or 1 in 1 million. And so Schrodinger argued that to achieve that level of accuracy in a classical system that was based on statistical mechanics would require billions of particles to be involved in this information transfer. And at the time, in 1944, people knew that genes were very small, and they were too small to contain billions of particles. And so Schrodinger thought there must be something else going on here. There must be some other description of what's going on in a gene. And what he did in this book was he proposed that a gene must be made up of some sort of aperiodic solid, or aperiodic crystal, he called it. And he said that these crystals should contain some sort of code script which is written at the single molecule level and whose behaviour is determined by quantum mechanics. So what Schrodinger was arguing in this book was that indeed quantum mechanics is at the very heart of biology. Okay, it's at the very heart of life and the way we transfer information. And was he right? So almost a decade later, Crick, Watson, Wilkins and Franklin discovered the structure of DNA. So this is the famous X-ray crystallography image that James Watson looked at, and straight away he knew that DNA was a double helix. And now the extensive work we've done beyond that shows indeed that DNA is made up of a linear sequence of non-repeating units, which can also be termed an aperiodic crystal. So this confirms what Schrodinger had originally thought. And what's even more interesting is that if you look at how the information is written on DNA, it's written on these base pairs. And the way that it's written is determined by the position of a single proton. So this, if you like, is a quantum code. This is information is written at the level of an individual atom. Okay? And this information must therefore be described using quantum mechanics. So there was a direct link between quantum mechanics and biology. And so this was all happening around the 1960s, but then there was a bit of a divergence, if you like. So physics basically went one way, genetics and biology went another way, and there was no real need in genetics or biology for quantum mechanics. They didn't need this complicated theory which predicted all this weird stuff. And they went on to map the human genome. Now we can go in and edit DNA as we like. So there's been a whole bunch of new and exciting explorations in biology. And what have we done in physics? Well, I won't claim that we've done all of this, but there was what we call the first quantum revolution. And having a thorough understanding about how quantum mechanics works, the physicists then went on to look at the applications of quantum mechanics. So what can we build? What devices and technology that can we build that relies on quantum mechanics? And so some of that technology that came out were lasers. Using quantum mechanics, we learned how semiconductors worked, and that enabled us to build the first silicon chip, which is now in our PCs, which drives our iPhones. MRI machines are governed by the principles of quantum mechanics. Okay, all of these technologies, PCs, atomic clocks, all use quantum mechanics to operate. Okay, this is what we call the first quantum revolution. Okay, and so the question becomes, is there ever going to be another convergence of quantum mechanics and biology? And so Philip Ball in 2005 wrote this article in Nature where he declared the dawn of quantum biology. And in this paper, he talks about some of the work that was going on around the world where people were looking for quantum effects in biological systems. And so in this paper, he outlines where people think quantum effects may be happening. And so one of those is photosynthesis, the other is magnetic sense in birds, enzyme reactions, the sense of smell we have, and possibly genetic mutations have all been linked in some way to some type of quantum mechanical effect. And tonight I'm just going to touch on the first two of these. And so the first one, we have probably the most evidence of quantum mechanical effects happening, and that's to do with photosynthesis. So photosynthesis is the process in which sunlight is incident on a plant, bacteria, whatever. That sunlight is absorbed by what we call antenna pigment molecules. So these are chlorophyll molecules. And what happens here is the photons from the sun come in, get absorbed by these chlorophyll molecules and create what's called an exciton, which is an electron hole pair. And what happens then is this electron then needs to find its way to what's called the reaction center. And when the electron arrives at the reaction center, all of that energy from the electron is dumped into the reaction center, it's converted into chemical energy, that's then used by the cell or the bacteria to make sugars, to convert CO2 to oxygen, for example. Okay, this is the process of photosynthesis. Now, if you were to think about this system classically, okay, if we describe what's happening here classically, the electron needs to hop from chlorophyll to chlorophyll molecule to make its way to the reaction center. And it can do so in a variety of different paths. And every now and then you're going to lose an electron because it's not going to make the reaction center. And also, as it does these hops, it's going to lose a little bit of energy. So you're going to lose some energy to heat, for example. And so when people started looking at this system, what they wanted to know was how efficient is it? How much of the energy that's absorbed from the photon makes it to the reaction center? And what they found was that this process is almost 100% efficient. So 100% of the energy that's absorbed by this photon is transferred into this reaction center. And so if you were to describe this classically, that simply wouldn't be the case. That was the first thing. The second thing was that if it takes some sort of random walk to get to the reaction center, that would take a certain amount of time. And you can work out roughly what that time would be. And the time was on the order of several nanoseconds. And then when they actually went and measured that, this process was happening much faster. The electron, as soon as it was produced, was getting to the reaction centre much, much faster than via a classical route. And so people started thinking about whether there were quantum effects at play here. And so one of the things that was proposed was that if this electron can remain coherent through this process, then it can be, if you like, in a superposition of these states. It can explore all of these paths at once and identify the most efficient path to the reaction centre. Okay? If it's in this quantum superposition, it must be coherent. Okay? That was the key requirement. And people doubted this very much in a biological system. So in biology, it's very wet, it's very warm, you've got lots of vibrations and complicated things going on. And so people didn't expect to see quantum coherence in a living system. Okay, that was the downer on the theory. But in 2007, some people used some very clever spectroscopy techniques to actually show these are quantum beats between these electrons. And if you're getting quantum beating, okay, this is similar to that interference pattern that I was telling you about before. If you get quantum beating, these electrons must be coherent. Okay, and they're coherent on the same time scale as it's taking the electron from when it's made to get to the reaction centre. So this is very strong evidence that photosynthesis indeed uses these types of quantum effects to its advantage to make this process 100% efficient. And so if we think about where we could go in the future, can we look to biology to understand how it does this so efficiently? So at the moment, when we think about our solar cells for clean energy, these are running at about 20% efficiency. Okay, is it possible to engineer cells based on these sorts of principles and get that efficiency up to 100%? Okay, that's the dream. That's one of the things that people are working towards. Okay, the other thing that people are looking very hard into is something called the magnetic sense of birds. Okay, and so let me tell you about that. So birds are known to migrate, and they can migrate over very vast distances. And so this is a map from Cornell where they mapped 180 different species of birds, and this is their migration pattern over the entire year. And when birds migrate, they migrate from one continent to another continent. And the question is, how do birds do that? How do they navigate? And they end up in exactly the same place in a continent up here every year with extreme accuracy. How do they do that? We know that they've got a visual sense, we know they've got a sense of smell, but they must have some other navigational ability that lets them travel such huge distances without getting lost year after year. And so in the 1970s, there was a couple, the Wiltsch-Quos. They did a very simple and elegant experiment to demonstrate that birds are indeed sensitive to magnetic fields. And so what they did back in the 70s was they captured one of these European robins during the migration period, and then they put it in one of these little boxes. Okay, and at the bottom of the box, there was a bunch of ink. So the bird would go in and then it would end up with ink all over its feet. And then there was a bit of paper that was put around this funnel. And what they wanted to know was which direction the bird wanted to go. And so the bird would try and fly out, and then it would fall back down into the ink pad. And then by looking at the paw prints when it was flying out of this thing, you could identify which direction it wanted to go. And so what they saw was in springtime, all these paw prints were pointing north. And in the fall, when they're flying home, all the paw prints are pointing in the south. Okay, now this, you could argue that the bird just knew where it was going and it always wanted to go up and it always wanted to go down, for example. Then what they did was they added some magnetic coils to this experiment where they could cancel out the Earth's magnetic field and introduce their own magnetic field. And when they do that, they can steer this bird into any direction you like. So you can steer it left, and so it thinks it's going north, but it's actually going east and west. So it was clear that the bird was responding to magnetic fields. Okay, these days there are much more sophisticated behavioural studies to show that this is indeed the case. So categorically, we know that birds use the magnetic field lines of the Earth to navigate. What we don't know is how they do it. And this is one of the interesting things. And so at the moment, there are three models that talk about how these birds are able to detect these magnetic fields. Now the first one, and this is probably the most popular one, is that somewhere in the eye of the bird there are these proteins called cryptochrome. And these proteins absorb light, so they need very small amounts of light, so because birds can still navigate in the dark. They need small amounts of light. Light comes in, incident on these cryptochromes. When that light gets excited, that cryptochrome protein produces a radical pair of electrons. Okay? That just means there are two basically unpaired electron spins, what we call them. Now one thing I haven't talked about tonight is entanglement. And when this reaction happens, these spins happen to be entangled. Okay? And if they're entangled, it means that their fates are intertwined with each other. And it's a very hard thing to engineer, this entanglement. But the first question they had to ask when they were doing this research was, is this chemical reaction that happens in cryptochrome, is that at all sensitive to the Earth's magnetic field? The Earth's magnetic field is really weak. So there's very few things that are disturbed by the presence of the Earth's magnetic field. It's maybe 50 microtesla or something like that. And so the chemists started doing work on these free radicals, and indeed they found that the chemical reactions and the product of the chemical reactions are sensitive to changes in the direction, the strength and the amplitude of the magnetic field. And so that's one of the first important points ticked off. The second was, if you create this entangled state and then you expose the bird to some particular form of electromagnetic radiation, you should disrupt this entanglement. And you should be able to flip one of these spins as opposed to the other. And if you do that, then you're going to disrupt the bird's ability to navigate. And so they did behavioural studies where they applied megahertz radiation, and indeed the birds lost their ability to navigate. So that again suggested that this process has some sort of role to play. But the outstanding thing for this model to work is that these entangled electron spins need to remain entangled for timescales up to microseconds. And so for photosynthesis, what I showed you before, quantum coherence was lasting for hundreds of femtosecond. Okay, now we're asking this entanglement to last for 6 orders of magnitude longer than that, okay, 1 million times longer. And entanglement is one of the really hard things to engineer. So the way physicists do it is we cool everything down to absolute zero, we try to control as much of the environment as we can, and this model is now relying on this entanglement existing in the retina of a bird with all this complex stuff going on at 37 degrees. So that's its main objection at the moment. The other one is that the birds have some sort of magnetic particle. They have like a little compass, if you like, in them. So you've got to look for these magnetic particles that exist within birds. And so in 2007, there was a lot of excitement about the discovery of magnetic particles in the beak of homing pigeons. And so this was great. So there was magnetic particles in the beak. These particles were sensitive to the Earth's magnetic field. They were located near known neuronal pathways. So if you want to find a mechanism for this system to work, a change in a magnetic field has to then get back to the brain of the bird, okay, to tell it to change direction. So that was fine, there was no neurons there. But latest research says that these ion clusters that were found are just to do with ion storage and not to do with magnetoreception. And that was based on the fact that these were found in random positions all of the time, and if it was in some sort of sensory mechanism, you'd expect it to be in the same locations for each bird or each species. So this is now a little bit defunct. The latest one is to do with magnetic particles existing in the ears of pigeons. So people have done some work looking at the brain activity of pigeons and their response to magnetic fields, and then they found out that if the inner section of the ear was removed from a pigeon, that its brain activity was reduced. And so my brain activity would probably be reduced if you cut my inner ear out as well. But people started to look in the inner ear for magnetic objects, and indeed they found these little blue dots that you can see here are very tiny iron granules. And what's peculiar about these granules is that there's only one per cell. So you'll see there's one in that cell, one in that cell. They're in about 30% of the cells, and they're located just under the hair cells of this pigeon. Now, the inner ear is a known sensory mechanism, so it's responsible for balance, for hearing, etc. So we know that there are pathways to the brain. And so what we know so far for this theory is that there are iron particles, and that's because if you stain with a particular stain like they have here, if something comes up blue, it's iron. Okay, so they know it's iron. It's located near known neuronal pathways, but is it sensitive enough to respond to the Earth's magnetic field? That's the question. And to answer this question, you need a magnetic field detector that is so small that it can detect the magnetic field from a single granule, which is 400 nanometers in size. Okay, that's the scale of this challenge. So we want to ask the question, how magnetic are these particles? And so what I want to show you tonight is some of the work that we're doing here at the University of Melbourne to address this question. And the way that we're doing it is we're creating these magnetic field detectors and imaging systems based that operate and exploit quantum mechanics. Okay, so I'll talk a little bit about that in a second. And so this is now leading to what we call the second quantum revolution. This is quantum 2.0. And this second quantum revolution is about not just building technology that's underpinned by quantum mechanics, it's building technology that uses quantum mechanics. So this year, Michelle Simmons from UNSW was awarded Australian of the Year for her work, pioneering work, trying to build a quantum computer atom by atom from the bottom up. Okay? If this works, this quantum computer takes advantage of things like superposition, entanglement. It uses these quantum mechanical effects to its advantage so it can process information faster, for example. We're also using quantum entanglement to create ultimately secure communication links. And what we're doing here at the University of Melbourne is we're trying to build improved sensors. Sensors that are so sensitive they can tell us new information about biological systems. And we want to build these sensors and start applying them to biological systems to understand things like magnetoreception in birds, for example. And so the dream, this was an image by one of the PhD students in the group, Victor Perunicic. And the dream here is to have some sort of quantum probe which can tell us the position of individual nuclei within a biomolecule, for example. And so this is basically like a molecular microscope. And if you can build one of these things, you'll have drug companies beating down your door because they want to know how drugs act on things like single biomolecules, ion channels, things like that. Okay, so the idea here is to build technology that uses quantum mechanics to tell us something new about biology. And so the way that we're doing that is with the material which is diamond. So it's not cheap, it's reasonably expensive on some of the things that we use. But it turns out that diamond have these amazing defects in them. So if you actually look inside a diamond at the atomic structure, if we kick out 2 carbon atoms out of a diamond, we replace one of the carbon atoms with a nitrogen atom, and we leave the other side vacant, we can create what's called an NV defect. Okay? And these days we can engineer this. We can do this into diamond almost at the single atomic level. Okay? We have that level of control and precision. Our diamonds don't look like this. Okay? Our diamonds look more like this. And these days we can grow diamonds. So we're not relying on natural diamonds that we mine from the ground. We can actually grow this material. And we grow it in things like a microwave oven. Okay? It's not too dissimilar from this. Okay? We put some gas in. We've got a microwave. We get a plasma in our microwave oven that looks like this. And we can grow diamond layer by layer and then insert these defects as we like, either during the growth or using techniques that David Jamieson works on, such as ion implantation. Now what this does for us, it gives us a beautiful single quantum system that we can play with. And the nice thing about diamond is that this quantum system works at room temperature, which is one of the most amazing things about it. So because it's in this robust carbon cage, we can have a single quantum system operating at room temperature. And so what can we do with that? What are some of the things that we can do? So this quantum system can be described very simply. So this is not all of the behaviour, but it can be described simply as just two levels. Okay, so this is here. I'm just looking at the ground state of this this defect, and so there is a bright energy level and there's a dark energy level. Okay, I'm calling it. And the nice thing is, if you interrogate diamond with light, if I shine a green laser pointer like this on this defect, that defect will fluoresce, it will glow, it will give me red light back. Okay? And it turns out that if I shine the green light on for long enough, I can put all of the electron spins into this level here. I can polarize all of the electron spins. This is the second amazing thing about this system. There are very few systems that you can polarize an electron spin at room temperature just with light. Okay? Normally you need massive magnetic fields, or you need to go to really cold temperatures to polarize electron spins. Here we get that for free. So I can put all of the electrons here, and if I want to, I can move the electrons from this level to this level if I provide just the right amount of energy. Okay? If I apply the right frequency, I can transition the electron from here to here. And when I do that, the amount of light that I get back from this system is reduced. Okay? So that's how this quantum system, that's the simple version of this quantum system. So what we have is the ability to initialize a quantum state. Okay, so I do that by just shining this green light on. We have the capability to read out the quantum state, and we read that out based on the amount of red light that we get back. And what we want to do here is we want to expose these delicate quantum states to some sort of environment. Okay, and the environment that I'm going to demonstrate here is a magnetic field, and it can also be temperature is another example of the environment. And so depending on the environment is either going to be kind to this quantum state or it's going to be nasty to this quantum state. And if we can monitor the quantum state, then we can understand things about the environment. That's what we want to do. Okay? So that's the principle of this quantum sensor. All right, so what I want to do is demonstrate that this is not science fiction. So quantum technology now is at the point where we're using these systems in our lab routinely, and we can come to a public lecture, and I'm pretty confident in saying this. This is the first demonstration of a quantum type measurement in a public lecture that's ever happened, if I can get it to work. All right. Now where's all my... So what I want to do is, hopefully that'll work. Very good. Okay. So this is my little laptop here. I'm going to turn on my green laser. Okay, so for the people in the front row have got the best view of this system. I'll show you, what do I want to do? Let me show you the other camera first, just to show you what's going on for the people at the back. It will come. There we go. Okay, so what I'm showing here, I've got a diamond, and you can come up for a closer inspection if you like after the lecture. I've got a diamond here. I've got a green laser that's coming onto this diamond. I've got a little microwave resonator here, which I'm going to use to try and find out where the energy levels of this diamond are. And I've got a little detector here, which is collecting that red light. Okay, and what I want to do is I want to probe these energy levels in this diamond. I want to probe the energy levels. We're looking at probably 1 million quantum sensors in here at the moment, and I want to know where these energy levels are sitting within that quantum sensor. So what I'm going to do is I'm going to monitor that red fluorescence, and then I'm going to scan a microwave frequency using my little resonator here, and when I hit the right frequency, I'll transition the electron from this 0 state to this plus or minus 1 state, and I'll get less fluorescence. Okay, that's the idea. So let's see if it actually works. Okay, so here, let me get that out of the way and I'll run my little program. And hopefully, oh, hang on. Okay. All right, yes, we're away. Okay, so this is in real time here, so I'm not doing any tricks. This is no magic. And what we can see here is if I monitor the intensity of the light that's coming from the diamond and I scan the microwave frequency, there's a point where the fluorescence, the amount of light that's coming back from this diamond, changes, okay? So now I know precisely where these energy levels are. All right, and in the diagram here, you can see if I've got no magnetic field present, these two energy levels overlap. And if I apply a magnetic field, okay, what I should do is split these energy levels. Okay, this is called a Zeeman splitting. So if I apply a magnetic field to this diamond, I should see the energy levels from the quantum sensors inside the diamond split. Okay, doing this measurement. So I've got here a little magnet, okay, on this stage here. And if I bring this magnet closer to the diamond and stop wobbling it, maybe that's too close. Let me go back a bit. Okay, you can see that line has now split from 2 to 4. And if I bring the magnet a little bit closer, okay, that splitting's got bigger again. And so this diamond, just by watching where these energy levels go in this quantum system, I can tell you exactly the strength of the magnetic field. I can tell you the direction of the magnetic field, all using, oh, hang on, maybe I, is it? Lost my counts, there we go. Simply by watching these energy levels, okay? So this is a really amazing and unique thing about diamond. So the reason that there are so many peaks now is because this NV sensor that I talk about, it can exist in 4 different orientations. So it can sit within the diamond lattice in 4 different ways. And what that means is we can probe the magnetic field in 4 different directions. And so if we do that, then we can reconstruct the whole vector magnetic field if we want, using this type of sensor. And these sensors are incredibly sensitive to magnetic fields. And what we hope to be able to do one day is use bulk sensors like this to measure our brain activity. So our brain activity, the magnetic signal from your brain, is extremely small. It's in the order of femtotesla. And the way people do this at the moment is they stick your head in a massive cryostat to get these magnetic readings. Okay? What we want to do is move towards some sort of quantum sensor which is operating on the principles which I'm showing you here based on diamond, and we think we can achieve that. So that's one example. Now, let me see if we... How am I going for time, Lloyd? Am I okay? How long? 10 minutes, all right. Okay. So the other thing we can do, and I might show you this as I keep talking, is we can change this up a little bit and we can not measure magnetic fields. The other thing we can measure is temperature. So I better put gloves on here. Okay, I'll do the right thing. Safety first, safety first. Okay. So now if we, let me add the, you can't have a good science experiment without liquid nitrogen. So let me now put liquid nitrogen in. I've got a little well up here, and I was told that it's sealed. And so what I'm going to do is fill this up, and I'm going to cool down. I'm going to cool down this rod. It's bubbling away nicely. Okay, and what I want to show, better start measuring. All right, I'm going to zoom in now at that peak I had in the beginning. So now I've taken away the magnetic field. Okay, here it is. Okay, and here's what the peak is doing. And I'm just going to let that bubble away while it cools down for a little while. And I'm going to tell you about temperature. So what happens when you cool down a diamond? When you cool down, a different thing happens to these energy levels. So when you put a magnetic field on, it splits them. If you increase or decrease the temperature, what happens is you shift them. Okay, these peaks translate. And so this is because you've got electrons. So there's two electrons that make up this NV center. And so what happens when you change the temperature is you contract the diamond or you expand it, depending on whether you heat or cool it. And this contraction or expansion changes the interaction between these two electrons that make up this NV center, and that translates to a shift, a tiny shift in these energy levels. Okay, and so what happens over time, so the middle of that peak started off at this red line, okay, and as this thing cools down, we're going to get a shift in these energy levels, and because I'm cooling it down, they're going to shift to the right in this case. Okay, so this one type of quantum sensor can tell us something about magnetic fields, and it can also tell us something about temperature. Okay, that's what we're doing these days in the lab. Okay, so this is quantum technology for your eyes. So there you go. So that's the first ever demo of a quantum measurement. I'm going to claim it. I'm going to claim it. All right. All right, so I'll turn the laser off so that, again, safety first. All right. Now, what can we do with these types of sensors? Okay, what are some of the things that we could think about using this sensor for? And so, in our group, we've been looking at several applications of this. And, hang on, I'm just going to turn off my microwave amplifier. And it turns out that you can make these sensors in single crystal diamonds, so these big chunks of diamond, or you can crush a diamond up into very small particles, into nanoparticles. And you can add nanoparticles into biological systems. So we started off many years ago asking the question, can we get nanodiamonds into biological cells? That was the first question. Can we then do these type of quantum measurements, which I've just showed you here? And can we report anything useful from these sorts of systems? And so it turns out we can. So we've demonstrated we can get them into cells. We've used them to map temperature in primary cortical neurons. So the idea here is to look at neural networks and see whether the firing activity can give rise to temperature changes. And we're measuring that inside the cell. We've used it for magnetic sensing in cell membranes. So you can label these cell membranes with particular spin labels, and we can measure the magnetic signals from those. And using these chips, what you can build is basically a new form of microscopy. So I said before, we can engineer these defects anywhere we like. And so what we're doing now, we're taking these beautifully clear and thin diamond membranes, we're implanting these NV sensors at the very surface across the whole chip, and we're now building microscopes that can image magnetic fields. And so this is a magnetic image of a hard drive, and you can see the magnetic bits of a hard drive. And this was some work done by Jean-Philippe Tétienne in our group, who's imaged the current flowing in graphene. So this is a single atomic layer of carbon, and using this magnetic field sensing technology, we can image the current flowing in a single atomic layer. And some of the stuff I'm involved with is trying to use this magnetic detection technology to do chemical sensing and start to look at these reactions like what's happening in the bird's eye, for example. Okay, so this is some of the work that we've done. And if we go back to the pigeon, okay, now that we know that we've got this cool quantum sensor that we can play with, and we ask the question of, can we actually measure the magnetic field from a single ion granule with this sort of technology, this sort of quantum sensing technology? Okay, I'll skip these bit in the interest of time. This is what it looks like. So, I should have pointed out, sorry. So we're working with David Keyes. So David Keyes is in the Institute of Molecular Pathology in Vienna, and he's the one that discovered these iron granules in the hair cells of pigeons. And so he's provided us with these slices of the inner ear. And to give you an idea of the scale here, so the distance between this black resonator here is 1 millimetre. Okay, and if we zoom in, we can make out poorly areas where there's cells, and if we keep zooming in, there are these 2 iron granules here and here. And those at the back will have no chance of seeing what that looks like. Okay, but believe me that they're there. And if you go and look at these in an electron microscope, this is what you would see. So here is a single cell with a single iron granule in it. And when we put this onto these sorts of diamond magnetic microscopes, we can indeed get a magnetic image of this single iron granule. And so this is work done by Julia as part of her PhD. And this is incredibly exciting, and so Liam's also been doing theory to understand the magnetic properties of this magnetic particle. So this is definitely the first magnetic detection of a single iron granule in the inner ear of pigeons. But the big question is it magnetic enough to respond to the Earth's magnetic field? That we don't have the answer to yet. Okay, that requires many more studies. We need to look at many more particles, and they should come on Tuesday. So if this, if the lecture was a little bit later, I might have some answers. Okay, and the last thing I want to touch on is, so there's a general theme here about, I think, how we can learn from biology to do things better ourselves, whether that be build better technology, engineer different materials. And one of the other interesting projects that we have at the moment is looking at these sea mollusks. These are called chitin, and so you'll find these in the rock pools out in Queenscliff, for example. And the interesting thing about these guys is that they sit on rocks and they eat the rock to get algae. That's how they survive. Okay, and they just move around on the rock, eating rock all day. And so to do that, if you want to eat rock, you need hard teeth to be able to do that, right? And so these chitin, their teeth look like this. So it's this long conveyor belt, and they mineralise their own teeth. They make their own teeth. They wear out one every 2 days. And this, from a biological point of view, this is a beautiful system to study this biomineralisation process. You can, in one set of teeth, you can look at the whole process of biomineralisation at once. And so it turns out that the teeth that they have at the very end that they're using to scrape on the rock are made out of iron. And so they have magnetic teeth, these things. And this form of magnetite, which they make at the very end, is the hardest biomineral that people have found, people have identified. And so to compare it to other things, it's harder than stainless steel, it's harder than an abalone shell. And this animal is able to do it at 15 degrees just by getting iron from its surrounding and assembling it in a particular way. And if you were to ask a chemist to deliver this sort of material, they would need to do that at hundreds of degrees. They'd need acids and bases to deliver the same sort of magnetic material. And the material that they do produce is inferior in terms of the hardness. So this animal is somehow able to do this in a much better way than we understand at the moment. And the way people study this traditionally is you go out to the Australian synchrotron, you book your time, you book a week out of your life, you go and do these measurements, and you'll find out there's regions where there are magnetic elements present, and you get these sort of maps. Okay? These maps are great, they're very instructive, they tell you what elements are involved, but they don't tell you how this animal is able to assemble this iron in any real way. And so we're starting to use our diamond microscope again to look at the properties of these things. And in particular, we're interested in when the mineralisation changes from one form of iron to another. So this orange that you can see in this image is the iron changing from ferrihydride to magnetite. And that's the most interesting part of this process, what happens there. And so doing that on our diamond microscope, we can produce beautiful magnetic maps which rival what you get out of the synchrotron. But the nice thing is, I said before that we've got these 4-axis that we can look at. So we can build up much more detailed magnetic maps, and we can actually have a look at how the magnetic domains are arranged in the teeth. Okay, and so what we can do is build up these really cool images which are showing you the direction and the strength of the magnetic field in these teeth. These teeth are about probably 0.5 millimetre in size in total, okay, and we're getting very, very detailed information about how these magnetic domains are assembled. And this information will certainly help the biologists and the chemists understand how the magnetic assembly is actually happening. And you can see here that we get these very detailed magnetic maps. We can tell you where there's regions of ferrihydride, where there's regions of magnetite. And so this is providing the biologists and chemists with information that they haven't had before. And that's what this quantum sensing technology is all about. Okay, so to finish up, I've got a couple of slides, Lloyd. I hope I'm not going over time. So here I've pulled a roadmap from the Quantum Flagship, which was a big European funded initiative, $2 billion. And this is their roadmap of where quantum technology is going in the future. And so you can see at the moment, here's our diamond sensor. That's actually a picture from a diamond paper. And so we have this technology now. Okay, this is ready, we can deploy it, we can start asking and answering interesting questions with it. Where we're going in the future is communication systems based on quantum entanglement, things like quantum simulators, the quantum internet, and ultimately the quantum computer. What's not on this map is how quantum and biology will come together, and I think that is a missing piece that hopefully we can exploit here in Australia, because I think we can learn a lot from biology which will actually help the quantum program here in Australia. And if we look around the world at what's happening in quantum technology, this is a very exciting time for the field. So at the moment, so I mentioned this flagship program, which was a $2 billion Euro investment in quantum technologies. China has just committed $10 billion US to build the largest quantum city, which is due for delivery, I think, in 2 years. The UK have put £270 million into their quantum technology program. Here in Australia, we've got 2 centres of excellence centred around this with relatively modest funding compared to the other countries. And you can see Singapore, Japan have also heavily invested in quantum technology. So this really is the way of the future. Companies like Google, IBM, Microsoft are now all have dedicated quantum technology programs existing within their businesses. So this really is an exciting way to go. And for all the young people in the audience, I think quantum technology has a very good future. So with that, I want to acknowledge the team that I work with. So Lloyd, and this is Lloyd's group here, and all the people in green. I spoke about their work tonight. I want to thank you very much for attending, and I hope you've learned something interesting tonight. I'm pretty happy when you.
I think we have time for just a few questions.
Not from my son. We'll answer that at home. Yeah. No. Okay.
Thank you, lovely lecture. How much quantum technology help us to understand how consciousness and mind work by matter?
To be honest, I don't know so much about this this field, but I understand that there are ideas around quantum consciousness and things like that. To be honest, that enters more into like a philosophy debate rather than something that I can measure. So I'm very interested in things that I can measure and prove, and I haven't yet seen a scheme in which we can experimentally determine questions like that, I think. Yep.
Yes.
So, lovely lecture, so really liked it. One quick question. So far we don't know why we sleep these days, so will quantum mechanics actually determine why we sleep, or give us a clue of why we sleep?
Again, I don't think that's a great question. I'm not sure. Certainly we want to use this technology, as I said, to start to monitor brain activity and understand how we think and how we process thoughts. Where that will take us, I'm not sure, but maybe that's one possibility. Yep. Yeah, great question. So the question was how close can we get these sensors, or what's the density of these sensors in the diamond? And the way that we make these defects is we accelerate nitrogen ions into the diamond. And so depending on the acceleration energy, that depends on how deep these ions go, so we can control the depth. And then in terms of the density, that's determined by how many we throw at the diamond. And so we can get these things to within about 10 or 20 nanometers, so very, very close together. And that's what we need to do this microscopy. We want to have all of these defects within a single imaging pixel. We want to have all four orientations and as many of these quantum sensors as possible.
Is that flat, or can you do that in 3D? Are there 3D sensors, or do you use flat ones?
Yeah, so at the moment we're doing 2D. And so we implant them very, very close to the surface, so they're about 10 nanometers deep. And then we put all of the interesting stuff on top of these diamonds. But if you want to do something like measure the brain activity, then you want as many of these sensors as you can get. So then you want to create these defects the whole way through that diamond and use all of them to your advantage. Yeah.
There's one over there. Had your hand up for a while.
Yep.
Any comments about what the Penrose's hypothesis about these microtubules having quantum effects inside them having something to do with consciousness?
Yes, so this is Penrose's idea about microtubules having an impact on consciousness. I don't have a view. I've had a look at a couple of the lectures. They're certainly interesting. I think the idea that microtubules can in some way process information, but I haven't seen experimental evidence or proof to say yay or nay. It's certainly an interesting theory. Yep.
If entangled particles can be separated by great distances,
Mm-hmm.
is the information being transferred faster than the speed of light?
It's a good question, and we were having this conversation the other day. So no, it's not, and it's because you're not transmitting information. So when you separate these particles are correlated. So whatever happens to this one will affect this other one, and that happens instantly. Okay, but you're not transmitting information. When you do this type of measurement, the outcome is random. So there's no information that's actually being shared or processed. If you want information, you need to tell one guy at the other end what your measurement basis was, for example. So, yep.
And there.
Yep.
Yeah, given that everything that the human body is made from has been successfully described by the standard model of particle physics,
Sure.
so that makes this sort of like biology work with quantum mechanics a no-brainer, doesn't it?
Yep. Oh, yeah. Sure.
It's getting from that to the reality.
To, yep. Yep.
Look, I think we might leave it there. If you have questions, come down afterwards.
Sure.
But don't get too close to the equipment, especially the liquid nitrogen. You know, that's the
But I've got some diamonds here if you want to have a look at the diamonds that we make these days. If you want to donate any diamonds, I should say, feel free to make any kind donations. That'd be much appreciated.